Hi, everyone, and welcome to part 2 of our lecture on Lab 11. In this video, we're going to cover the urinary system component of this week's lab. So let's get started. Now, the urinary system we've already discussed in lecture. Remember, this is our excretory system. One of them also has a very important role in regulation of contents of our blood and our water volume. Now, this system is composed of two kidneys. Remember, they're retroperitoneal, so they're outside of the peritoneal cavity. They each have their own ureter, which is going to drain the urine down to the bladder, which is removed from this particular model. And then you will exit through the urethra. So the goals for this week are to not only be able to recognize these major structures and these major organs and the structures within them, but also to identify several structures within the pig kidney as well. So here is a frontal section of a kidney. And remember, the kidney has multiple layers to it. So we have an outer region called the cortex, a middle region that's called the medulla, and then right down here is going to be the renal pelvis. So the important thing to remember in terms of lab is when we are identifying, if I say what is the region, that is going to be medulla or cortex, right, with these eye bars. If I'm looking for structure, like say the renal pyramid, that would be more of what is the structure. So again, reading the question will give you hints as to what exactly I'm looking for. Now, again, we have urine formation that's happening both in the cortex and in the medulla. And when the urine's produced, it's going to exit the pyramid through the minor calyx and then flow into the major calyx. And then these major calyces then fall into the renal pelvis and then out of the kidney through the ureter. So where are we actually getting the stuff to make the urine from? Now, remember in lecture, we went through the entire system of vasculature, and we are going to learn them here in lab. So now we get that ability to take what we've learned in lecture and apply it to models in lab. So again, we're going to have paired vessels. Remember, the only difference here is in the venous system. Instead of having a segmental artery that runs sort of through the pelvis until we meet the renal column, we have only interlobar. So we go from cortical radiate veins to arcuate veins, and then interlobar all the way through until we hit the renal vein. So that's a really important one to remember. You're going to be able to visualize all of these on models, and we're going to be able to see some of them depending on how the pig kidneys have been dyed and cut. We'll be able to see a lot of them there as well. Now, in terms of vasculature, one thing I really like to emphasize is the distinction between the peritubular capillaries and the vasa recta of the nephron. So remember, the nephron, which is the set of yellow tubes here, that's what's making the urine. We have blood vessels coming in. So we bring blood in through the afferent arteriole. It's filtered through the network of capillaries called the glomerulus. And then whatever doesn't get filtered will exit Bowman's capsule through the efferent arteriole, which then branches and surrounds both the tubules in the cortex and the tubules in the medulla. So the tubules that are in the cortex, these are the ones that are going to surround the proximal and distal convoluted tubules. They are the peritubular capillaries. The ones down here that are going around the loop of Henle, those are called the vasa recta. And the reason I really emphasize the difference here is physiologically, they have very different roles when it comes to urine formation. So for example, the vasa recta is involved with the countercurrent multiplier, which is going to help create that concentrated urine. The peritubular capillaries have a different role in that particular aspect of the kidney. So I really like to make that distinction. So when you get into physiology, you already know they're two separate identities of these capillaries. Now the nephron, as we discussed, is where we're making urine, and it's divided into different regions. We're going to have the renal corpuscle, which is where we start our filtration at. We go into the proximal convoluted tubule, down and then back up the loop of Henle, into the distal convoluted tubule, and then to the collecting duct. So this is actually a zoomed in view of part of one of our models where you can see these multiple nephrons, right? So we have the renal corpuscle, which is where we have Bowman's capsule and the glomerulus together as one. You can see the different tubules and the different regions of the loop of Henle that then all connect to the collecting duct. So we have two of these models, and they're very good for kind of seeing the whole picture. And I'll show you guys at the end of the lecture some other models that we'll be able to use that are really helpful if you're still having some trouble orienting yourselves in terms of where things are in the nephron. Now, after we exit the kidney, we're going to head down to the bladder through our two ureters. And the ureters will then drain, they'll go behind the bladder and kind of insert on the posterior inferior aspect and basically fill the bladder up. The bladder, or urinary bladder, as we see here on the bottom, this is our storage site for urine. So it will fill up, and then we have stretch receptors that'll let us know it's time to void. Now, how we do that is by both involuntary and voluntary mechanisms. So we have our detrusor muscle that will help squeeze the urine out. And then we have our sphincters that are going to help relax and then allow the urine to pass. Now, because the bladder is a storage site, it's going to be able to expand. And so when you look at the inside of the bladder, you're going to see rugae, just like we saw in the digestive system in the stomach. The rugae, along with the transitional epithelium that we see up here, that we observed during our first lab, these are what allow your bladder to stretch. So it's a combination of both of these. And in lab this week, we are going to be reviewing the bladder. So we're going to be seeing this transitional epithelium again, but we're looking at the bladder from not as close up. So we're also going to see things like the lamina propria, which is just underneath it. And you're going to get to see that detrusor muscle as well. So here is a frontal view of a female bladder and urethra. This is not a model that we have, but it's a really good one for kind of talking about the exit of urine. So we have these trigone right here that we can see. That's the internal urethral orifice and our two ureteral entrances. And when we're ready to void, this sphincter here, the internal urethral sphincter, will relax. And that is going to open up the internal urethral orifice and allow urine to start to go down the urethra. We have control over the external urethral sphincter, which runs through the pelvic diaphragm. And then that is what we're going to relax when we go to the toilet. What's really important to compare this week is to look at both the male and female pelvises so that you can see the difference in the urethra. So again, as we discussed in lecture, women, as you can see here, have a very short urethra, and it is separated from the vaginal canal, which you can see, which would be posterior here. So our reproductive and urinary systems are separated. In men, they have a penis, and the urethra is going to run all the way through from the bladder to the tip of the penis. So they have a longer urethra, and it is also shared by the reproductive system, which we're going to talk more about next weekend. Not weekend. Now, one important thing about the male urethra is because it is so long, it's actually divided into three regions. So you have the prostatic urethra that's going to run through the prostate. This is a male bladder. So here's the prostate. This would be the prostatic urethra here. We then have the penile or spongy urethra that's going to run through the penis. And then there's a smaller region that's about 1 centimeter long that we don't observe in this class just because it is so tiny. And that's called the membranous urethra. And that's the part that kind of is the transition from prostatic to penile as it runs through the pelvic diaphragm. So here are some of the models that we're going to be looking at this week. We have some of our kidney models that you might have observed during the vasculature lab. We also have Saxon as well, very good for looking at position of kidneys and ureters as well. And I have a couple of these 3-part kidney models that show you the frontal section of the kidney, very similar to the diagram we looked at the beginning, a zoom in of where the nephron is, and then a zoom in on the renal corpuscles. You could see the afferent and efferent arterioles. You could see the podocytes and pedicels of the glomerulus as well. On top of that, we have this guy here, which is our nephron model. This is the one where if you're still having trouble determining who goes where in the nephron, this is a really good one because it is color-coded. Now, in the previous slide with the 3-part kidney, that is also color-coded in terms of the nephron, but this one's just really big, so it makes it easier for you to visualize it. We also have our male and female pelvic models, so you can see the ureters and the bladder, as well as the urethra in relation to other pelvic organs. And of course, we're going to be looking at our pig kidneys this week. And as I mentioned, depending on how the kidney was stained, how we cut through it, we'll be able to see a lot of not only structures, but also vasculature. So that is it for our Lab 11 lectures, and I will see you guys in lab. Bye.